to welcome a diverse and engaged audience. Your participation greatly enriches our discussion and underscores our shared commitment to advancing in our respective fields. I look forward to productive conversations and collective learning as we embark on this educational journey together. SPSR is distinguished for its engaging webinars, further enhancing its status as a leading platform for continuous professional development in the pharmaceutical sector. I am confident that today's session will uphold our tradition of excellence, providing valuable insights from a respected speaker. Let us seize this chance to gain new knowledge, participate actively, and progress our field. Welcome, everyone. I look forward to a productive experience for all participants. Thank you for joining us today. We are gathered today to commemorate the 95th SPSR webinar on targeted nanoconstructs for the treatment of autoimmune diseases, a distinguished event hosted by the Society of Pharmaceutical Sciences and Research, SPSR. Under the esteemed guidance of Dr. Shashi Alok, National President of SPSR, the Society continues to play a pivotal role in fostering innovation and advancements in the field of pharmaceutical sciences. The SPSR is devoted to advancing the pharmaceutical field through a variety of strategies. Our mission is to organize conferences that support pharmacy-related initiatives and endorse programs tackling public health issues in India, focusing particularly on disease prevention. We make it a priority to recognize and honor pharmacists whose notable contributions drive progress in our field. At the heart of our mission is a commitment to spreading pharmaceutical knowledge, ensuring a steady and robust exchange of information about pharmaceutical practices and services. By striving towards these objectives, we aim to elevate the quality and impact of the pharmacy profession, fostering a proactive and collaborative community dedicated to excellence. It is our great privilege to introduce Dr. Manju Rawat Singh, Professor, University Institute of Pharmacy, Pandit Ravishankar Shukla University, Raipur, Chhattisgarh, India, as a distinguished speaker of the day. Dr. Manju Rawat Singh expertise greatly contributes to our event, providing substantial knowledge and insights. Her participation is certain to enrich our discussions and expand exploration of targeted nanoconstructs for the treatment of autoimmune diseases. We are grateful for her engagement and anticipate an enlightening session. It is our privilege to introduce Mrs. Monika Sabharwal as the convener for today's webinar, who also serves as the founder and National Secretary of SPSR and CEO SPSR Periodicals Private Limited. Her significant involvement and leadership in pharmaceutical sciences and research communities are indicative of her commitment to the field. We are very grateful to have her guidance in today's session. I am pleased to invite Mrs. Monika Sabharwal to warmly welcome our distinguished guest speaker of the day, Dr. Manju Rawat Singh. Over to you, Monika, ma'am. Monika, ma'am.
Thank you so much, Mrs. Rupam Sharma. Hello, everyone. I am Monika Sabharwal, the convener of today's webinar. And it is with a great pride and enthusiasm that I welcome you all to the 95th webinar organized by Society of Pharmaceutical Sciences and Research, SPSR. Today's session brings into a focus a topic of immense significance in the evolving field of pharmaceutical sciences. Target nanoconstructs for treatment of autoimmune disease. Autoimmune disease represent a major global health challenge affecting millions of individuals and often requiring lifelong management. One of the major challenges in treating this condition is achieving site-specific drug delivery that can suppress the immune responses without compromising the overall immunity. This is where targeted nanoconstructs are creating a paradigm shift, offering a precision, reduced systematic toxicity, and enhanced therapeutic outcomes. Nanotechnology based platforms are now being explored to deliver immunomodulators directly affecting tissues, opening a new avenues for the safer and the more effective treatments. We are deeply honored to have with us today an esteemed and respected name in this particular field, Dr. Manju Rawat Singh ma'am, Professor at University Institute of Pharmacy, Pandit Ravishankar Shukla University, Raipur, Chhattisgarh, India. Dr. Singh ma'am is widely recognized for her contribution to the nanomedicine and drug delivery research, and her work has significantly advanced our understanding of how nanotechnology can revolutionize therapeutic strategies for complex disease, especially autoimmune disorders. Her insights today are sure to enlighten us and spark a valuable scientific discussion. As we mark the 95th milestone in the SPSR webinar series, I take this opportunity to express the heartfelt gratitude to our community of participants, researchers, students, and professionals. Your active engagement and the continuous support have played a pivotal role in the success of our mission to foster accessible scientific learning and encourage interdisciplinary collaboration. Our digital footprint continues to expand rapidly. The SPSR WhatsApp channel has now over 16,000 followers, and our LinkedIn network has grown to more than 7,300 professionals. SPSR is becoming a trusted hub for the pharmaceutical knowledge and professional development. All of our webinars are of free of cost, and participants receive certificates that enrich their professional portfolios. We have also received overwhelming feedback, with the majority of the attendees reporting that SPSR webinars have contributed positively to their academic and professional growth. Before we commence, I would like to warmly welcome our National President, Dr. Shashi Alok, whose visionary leadership has been crucial in a present transformation of the SPSR. I would like to extend my heartfelt thanks for the significant contributions made by Mrs. Rupam Sharma, the co-convener of this webinar, and organizing committee members, Dr. Pradeep Gulani, Dr. Saurabh Kose, Dr. Sanjay Nagdev, Dr. Ankur Agarwal, Mr. Junaid Tantare, and Mr. Rishabh Banu. Their tireless efforts have played a crucial role in transforming the vision of this event into a reality. Join SPSR vibrant community of over 57,000 pharma professionals actively engaging across LinkedIn, YouTube, Facebook, and WhatsApp. Let's connect, learn, and grow with the top-tier webinars and exclusive content every time. Do not miss out this unparalleled opportunity to elevate your pharmacy practice. Stay updated by subscribing to SPSR YouTube channel and enabling the notification bell icon for instant alerts. Follow our journey in advancing the pharmaceutical sciences that will benefit the society overall. Once again, I welcome you all and thank you for being part of this ever-growing global learning community. Let us now begin this exciting session with a great enthusiasm. Please join me in welcoming our speaker, Dr. Manju Rawat Singh ma'am, as we delve into the world of nanotechnology. And it is a promising role in tackling autoimmune diseases. Your enthusiasm, active participation, and trust in SPSR drive us to keep going strong every time. Thank you and welcome you to the 95th SPS webinar. Thank you so much. Over to you, Mrs. Rupam Sharma.
Thank you, ma'am. I appreciate Mrs. Monika Sabharwal granting me the opportunity to present this webinar. And now we will start with a short introduction to the guest speaker of the day. Dr. Manju Singh is a highly accomplished academician and researcher currently serving as a professor at the University Institute of Pharmacy, Pandit Ravishankar Shukla University, Raipur, Chhattisgarh. With over 20 years of teaching and research experience, she has established herself as a prominent figure in the field of pharmaceutical sciences. Dr. Singh's research expertise lies in dermal drug delivery system, formulation of bioactive carriers using novel delivery platforms such as hydrogels, nanoparticles, and microspheres, as well as protein and peptide delivery systems. Her work also emphasizes topical lipid-based therapeutics for autoimmune disorders like psoriasis and rheumatoid arthritis and the application of computer-aided formulation techniques. She has an impressive record of academic output, including 128 peer-reviewed publications, 32 book chapters, and 3 published books. Her scholarly contributions have received global recognition with over 6,800 citations on Google Scholar and an H-index of 39, and 4,044 citations on Scopus with an H-index of 32. A recipient of several prestigious awards, Dr. Singh has been recognized in the top 2% of the scientists worldwide by Stanford University 2021 to 2024. And she has received honors such as the APTI Professor C.J. Shishu Award 2024 and the Dr. Ambikanand Mishra Award 2022. She is also the recipient of multiple travel grants, national research awards, and best teacher accolades. Her leadership roles includes serving as the president of IPGA Chhattisgarh branch 2022 to 2025, chairperson of the IPGA Women Forum, and former secretary of APTI Chhattisgarh. She has delivered 46 invite talks and played a key role in more than 7 government-funded research projects supported by DST, ICMR, UGC, and CGCOST, and others. With 5 granted patents, including 2 international, and ongoing collaborative research with institutions like IIT Bhilai, Dr. Manju Singh continues to inspire innovation and excellence in pharmaceutical education and research. It is with great satisfaction and pride that we declare we have exceeded 1,500 registrations for this webinar. Registrations received from India, Albania, Algeria, Bangladesh, Bhutan, Brazil, Ethiopia, Indonesia, Iraq, Kuwait, Malaysia, Nepal, Nigeria, Oman, Philippines, Qatar, Romania, Saudi Arabia, South Africa, Sri Lanka, Syria, Turkey, and USA, and Vietnam. We are deeply moved by the substantial and varied responses we have garnered. Our heartfelt gratitude is extended to each person who has taken a moment to participate in this significant event. Thank you all for joining us today. Let's make this webinar an insightful and enriching experience for everyone involved. We have our esteemed keynote speaker, Dr. Manju Rawat Singh, with us. Welcome, ma'am. Your insights would be highly valuable, and I would appreciate if you could present them in a speech. Over to you, ma'am.
Thank you so much, Mrs. Rupam, for such a modest introduction. First of all, I would really like to congratulate the entire team of Society for Pharmaceutical Sciences and Research, especially Dr. Shashi Alok, Mrs. Monika Sabharwal, and the entire team of SPSR for taking the initiative of teaching so many people, sensitizing so many people on so varied topics of pharmacy. So really heartfelt thanks to all of you and a big congratulations to so that I could hear it that so many people have registered, which is even more than 1,500, which is really a commendable job on the part of entire team of SPSR. So I'm going to share my screen. So please suggest me if it is visible to all of you.
Yes, ma'am.
Okay. See, the topic that I selected for my talk is on targeted nanoconstructs in treatment of autoimmune diseases. Why I have selected this particular topic? Because this is really at a boom, especially with the advent of COVID-19, and there's so many cases coming up where our immune system is directly involved. And but it doesn't mean that if I say that this is really a dynamic field of targeted nanoconstructs in the treatment of autoimmune diseases, because nowadays everyone is talking about the immunity, the immune system. There are various ways of ways to improve upon your immune system, but this immune system also needs a control, also needs a check, because sometimes it happens that it could lead to the autoimmune disorders or the diseases. So autoimmunity in a way where our immune system is not able to recognize between the self and the non-self. So, see, in our lifetime, we encounter a number of different kinds of foreign particles. These foreign particles could be actually present outside your body, or these could be inside your body. I'm saying this that these are foreign particles because they are considered as non-self by our body. Non-self in the sense that these are not considered as our own part by the body, and that is why our immune system comes into play, and it just tries to, work towards expelling these things out of our body. Uh, so, and next thing which is there, that is, so these which are present outside the body, they are known as exogenous antigens. And those which are present inside the body and considered as non-self by our immune system, they are the endogenous antigens. Endogenous antigens are like, if some virus particle gains entry inside our cell and it makes the changes in our normal cells. So in that case, those cells which have been, modified by these viral particles, or because of the cancerous, the tumor condition, if these cells undergo any kind of change, then they turn out to be endogenous antigens. So even, if we compare with the exogenous antigens, these endogenous antigens are even more, dangerous to us. So they need to be kicked out immediately from the body. So, before delving into the more details of this kind of systems, I just want to give an overview about the immunity and the immune system. So, if we talk about the immunity, there are two aspects of immunity. The one is the innate immunity, which is present in our body right from the time of our birth, and this forms our, the part of our innate immune system. The another aspect is the specific immunity, which is more directional, which is more specific, and this we develop with a time span. That is, it is actually more of adaptive type. Now, coming to the innate immunity, the major components are like skin and the mucous membrane, and this involves the first line of our immune system. These involve the physical, these are actually more of physical in type, and they are more physical in the sense that they won't allow anything to gain entry inside a cell by forming a barrier kind of a thing. See, I'll give an example of our house. Okay, if any intruder wants to gain entry into our house, it must has to, cross it must has to open the gate. The walls are there, the windows are there, the doors are there. So all these are actually the physical barriers. So in the same way, in our body, the skin and the mucous membrane, they form the first line of our defense system. Next, under this category is the formed elements. If we talk about the formed elements, these are more of a cellular type, like all the cellular components, they come into this. Our blood cells, like RBCs, the WBCs, the platelets, all these are the components under the formed elements. Now, the third thing which comes is the non-specific chemical defenses. And this actually involves all the chemicals which are actually secreted in response to any kind of invasion. So these chemical substances could be even, your tear, the HCl, which is present in our the gut, the gastric, the stomach. All these come under the non-specific chemical defenses. And the fourth one under this category is the inflammation and fever. So if we say the inflammation, it is actually the immediate protective response to any kind of the invasion by the infectious agent. And next to this is the fever. But these inflammation and fever needs to be kept in control. Otherwise, it turns out to be a condition of autoimmune disease. Rest, these are actually protective to us. Then coming to the specific immunity, which are more specific and directional, we have the first component as the lymphocytes, and they are more specific, means they are directed towards the specific kind of antigens. They further, divide into the T cells and the B cells. These are actually the form of lymphocytes, and they are also involved in the more specific kind of immunity, that is the humoral immunity, which is the antibody-mediated immunity, and the T cell mediated, that is a cell-mediated immunity. Then comes the most important part in the immune system is the antigen-presenting cells. If we talk of antigen presenting cells, see, they are somewhat like when someone comes to our house and we, it's not like that you won't offer them something. We offer them the water and the other, the things to them in the proper tray. So antigen presenting cells actually serve this purpose. They serve like a platter, and they then offer the antigens to the further the cells like the T cells for having the proper action against these kind of intruders. So this is about the general immunity. Now coming to the origination of these kind of cells. So they are the multipotential hematopoietic stem cells, which further divide into the myeloid progenitors and the lymphoid progenitors, forming the different kind of cells which are there in our body. So this includes the when we talk about the the thrombocytes, the erythrocytes, the mast cells, the myeloblast, further refining into the basophils, neutrophils, eosinophils, and monocytes. Now these circulating monocytes, they further evolve in the form of a macrophages, which are an important part of our immune system, and they are the major players acting against any kind of foreign intrusion. Then next involves the next is under the lymphoid progenitor category is the natural killer cell, and the small lymphocytes like the T lymphocyte and the B lymphocyte. This B lymphocyte on activation forms the plasma cells, which in turn are responsible for forming the antibodies against the specific antigens, which have gained the entry inside the body. So we can say that these WBCs in our body, they are actually the they are the kind of circulating the police inspectors or the soldiers who keep on moving through this bloodstream, and they are look out for any kind of intruder in our body, and when they find them, they just pick them up and just throw them out of the body by engulfing them, neutralizing them, and then excreting the rest part out of the body, which is non-toxic to us. So coming to the type of WBCs, which are actually the major players. There are 5 different types of WBCs, which have been categorized as the granulocytes and agranulocytes. Coming to the category of agranulocytes, we have neutrophils, which constitute around 50 to 70% of the blood cell types. And, they are actually white in color. And this is responsible for the whitish kind of appearance in case of the pus-killed cells. Then comes the eosinophils, which constitutes about 1 to 3% of our blood type. And these are responsible for allergic response and asthma, and in case of parasitic killing. The third one are the basophils, which constitutes around 1%, and they are majorly involved in the allergic reactions. Then under the category of agranulocytes, which are the major players under the class of the specific immunity. So first in this is the monocytes, which constitutes around 4%, and they on circulation then become the macrophages. Another one in this category is the lymphocytes, which constitute about 33%, and they are the one which direct the immune system relating to the specific immunity. So these are all about the different kinds of blood cells which are involved in the immunity. So we can say that we have an entire convoy, the convoy of people who are working together, forming an integral part of our immune system to throw the, any kind of intruder out of the body. These are like, the cytotoxic T cells, Then the B cells, which specifically recognize a specific kind of antibodies and throw them out of the body. The regulatory T cell, they are somewhat like, you have seen the traffic police. So how do they regulate, manage the traffic? So similar way, regulatory T cells decide where the cells have to, which cells have to be recruited and where they have to be sent. Like if they have to be sent for apoptosis, they have to be killed, they will be sent to the apoptosis center. If they have to be sent for the effector region, like the B cells, they have to be activated into the plasma cells. It will be decided by the regulatory T cells. So this is it. And the last one is the the helper T cells, which together with these major cells further help the immune system to aggravate the other pathways of the reaction. So this is on a lighter note. You can see over here that what is the work of macrophages and how these intruders, they run out of a fear because of the presence of these macrophages. So this is another view. This is a structure of a macrophage, how it is like. See, I just, the morphology of these cells are in such a way, because I'm saying this thing because it will help you in deciding in making the kind of nanoconstruct that you want to make up based on the kind of structure of our normal physiological system. Like if you see the normal RBG, how it is like a disc shape, regular, smooth structured. Why so? Because it has to do a particular function of carrying the oxygen from one part to the another part. So it has to have a smooth surface area so that it can move from one place to another. Next, if we talk about the platelets, because it has to be involved into the blood clotting, it has to serve as a plug. So its structure is somewhat like that. And if we talk about the WBCs, the lymphocytes, because they are involved in the protective mechanism, they have to engulf something, hold something, engulf it, and degrade it. So their structure, the outer surface is almost like that to protect from these kind of intruders. So you can see over here, this green color structure, it's of macrophage. So these rod-shaped bacteria are there. They get entangled onto its surface. Then here you can see how it actually, by means of the cytoplasmic invagination, this gains entry inside the phagocytic cell. They get merged with the lysosomes, forming the phagolysosome. Then they are digested upon, they are acted upon by the hydrolytic enzymes, and then they are taken out from the body. So this is how actually the phagocytes or the macrophages, they work. And this entire process is known as phagocytosis. So these are there is a balancing component between the antibody and the cell-mediated immunity. If we talk about the antibody-mediated immunity, which is also known as humoral immunity, here the two kinds of cells are involved, that is the B cells and the plasma cells. And they then actually further produce the antibodies against the intruding antigens. Another branch is the cellular immunity, where these T cells are involved, forming the two kinds, that is the cytotoxic T cells and the helper T cells. And they are more specific. As you can see over here, the knife, they are more specific, pinpointed at the particular kind of antigen. So this is about the balancing act between the adaptive and the innate kind of immunity. Now, this structure, I'm just giving an overview about the structure of the B lymphocyte. See, we all know that the B lymphocytes, they originate in the pluripotent stem cells, then they mature within the bone marrow, and they then express, when they mature, they express a specific kind of receptors onto their surface, which are actually the membrane-bound antibody molecules. So you can see over here, now each antibody molecule has a particular site which is specific for a particular kind of antigen. Here it is represented with the red sign. So once the moment this B cell comes into contact with this kind of antigen, it will immediately bind to it, and it will then neutralize it, degrade it, aggregate it, and destroy it based on the different mechanisms. So this is how actually the B lymphocytes, they work. So this is just an overview that I wanted to give you, that how actually they work. This is an overview of the humoral mediated immunity. Just going to keep it over here. So you can see over here, these are the B cells with the antibody molecules on their surface. So this red structure represents the antigen. How it acts? Once it recognizes, it binds to the antibody molecule present on the B cell. They are internalized, degraded. Then there are certain components of the cell membrane which bind to it, and these are known as MHC molecules, that is the major histocompatibility complexes. Then this complex then binds to this is a T cell, and then the T cell receptor then binds to it. So in a way, we can say here B cell actually works as the antigen presenting cell. That is, it is actually presenting the antigen through its MHC to the T cell. So then after binding to it, T cell gets activated. It releases its different mechanisms. Gets activated. Its various mechanisms gets activated. And then it releases the cytokines, which further converts the B cells into the effector cells. And these effector cells are what? Which are responsible for particular kind of response. And they then form the plasma cells, which produce the antibody against the particular kind of antigen that they have encountered. So in a way, this number gets increased. See, there is also a very interesting theory of clonal selection, which says that although we have never encountered any antigen in our life, but when we encounter any antigen for the very first time, our immune system is so well built off such that that antigen will come into contact with these B cells, and it will send the signal that more of such the plasma cells needs to be activated. So they then actually release the surge of these kind of antibody molecules against the particular antigens that they have encountered. So this is how they neutralize it. Then comes the next branch, that is the cellular mediated immunity, whereby in which there are two kind of T cells are involved, that is the CD8 plus T cells and the helper T cells, that is Th1 and Th2. One more thing, CD8 plus T cells are the one which are effective against the endogenous antigens, means the antigens which are there within our body. And helper T cell pathway comes into play when there are exogenous antigens, means antigens which are present outside our body. So what happens in this case is that the first thing, that is CD8 plus T cells, they directly kill the infected cell they come into contact with. Th1 T cell, they activate the macrophages for the defense, and Th2 T cells, they stimulate the B cells for the antibody production. So in a way, the previous one that I've shown you is actually the Th2 T cell activation. So this involved actually this T cell is actually the Th2 T cell. So this is the general structure of the T lymphocytes. You can see over here. The only difference between these two kind of, the T cells, that is the CD4 plus and the CD8 plus T cells, is based on the receptors which are present on their surface. So it is CD4 plus kind of a receptor on the T helper cell, which is known as CD4 plus, and this one CD8 plus having the CD8 receptor, this is known as a CD8 plus cytotoxic T cells. Now coming to your cellular mediated immunity. See, if we remember, the B cells, they have a direct surface receptors, these antibody molecules, which can directly bind to the antigen. But if you see the T cells, they do not possess these receptors. T cells, they need the antigens to be presented to them like on a silver platter. So that we have shown before, I've shown before. So this is presented to T cells by means of the antigen presenting cell, as you can see over here. So T cells, they cannot bind to the antigens directly. They need to bind to this by means of the antigen presenting cells. Then coming to the two major components of these APCs, that is antigen presenting cells. These are of two types. The one are the general APCs, which are basically the endogenous antigens, and another one are actually the professional APCs, which are the exogenous antigens, which deal with the exogenous antigens. So general APCs are like all the cells can be general APCs. They are involved in the antigen presentation. They involve the MHC class 1 molecules in this, and there occurs the CD8 plus T cell interaction and further killing of the cell. Whereas if we talk about the professional APCs, the professional antigen presenting cells are there, like the macrophages, dendritic cells, the more specific antigens, and in this MHC class 2 activation is there, further activating the CD4 plus T cells. Why am I explaining this? Because if you are thorough with the basic concepts of immunity, you are definitely going to make a system which will be more targeted towards the any kind of immune disorder, whether it is immune suppressive, immune stimulant, any kind of the hypersensitivity, any case involving the immunity. Now coming to the work of the general APCs, I've told you that in case of general APCs, CD8 plus T cells are involved. You can see it is any cell which is serving the purpose of APC. If, for example, there is any protein which is considered as non-self by our immune system, this will be acted upon by the inherent proteases which are present inside the cell, and it will be cleaved into the smaller fragments, forming the peptides. Then MHC class 1 molecules, they bind to it and take them to the surface. So now here at this point, T cells can easily recognize them, and the T cell receptor on the CD8 plus T cells, they can easily bind to this complex of MHC class 1. And then because it is the endogenous antigen, CD8 plus T cells will become active, and they further release the cytokines, further killing the cells bearing these kind of antigens. Then comes the work of the professional APCs, whereby CD4 plus TH1 cell is involved. For example, this is the case of a macrophage. It represents a macrophage. This is the protein, which is exogenous and antigenic to us. So it is first recognized by the macrophages. It is internalized, again acted upon by certain proteases, which makes the fragment of it, converts them into the smaller peptides, and some component of the MHC1 binds to it, takes it to the surface. These are MHC class 2 molecules. And here comes the work of the T cells, where the receptors of the T cell, CD4 plus T helper cells, they bind to it, they get activated and release the number of chemokines and interleukins like IL-2, interferon gamma, TNF alpha, which act on these macrophages and directly kill them. So this is involving the CD4 plus TH1 cells. Then comes the work of the CD4 plus TH2 cells, which we have already discussed. That is the B cell with the antibody molecules onto their surface. They get internalized, bind to the receptors present on the B cells. The component of this MHC binds to it, takes it to the surface, and TH2 cells then then binds to it through its receptor, gets activated, and this in turn releases other interleukins like IL-5, IL-6, 10, and 4. And this actually converts this B cell into the effector cells. Now these effector cells means they will form the plasma. They'll get converted into the plasma cell. And these plasma cells then release the antibodies directed against the intruding antigen molecule. And then it is then free to move in the circulation to bind to all those antigen molecules, and then does its function. So there is a fine line between, the self and the non-self in our body. And immune system only reacts to the antigens which are considered as foreign or dangerous to us, or which is not a part of our body. But sometimes it might happen that there are certain infectious agents which, by means of molecular mimicry, they mimic some of our component, and our immune system mistakes that component as actually non-self, and then starts to work against our own body components. So there arises the issue of the autoimmune reaction. That is, our immune system interprets the body's own tissue as foreign and produces the antibodies called the autoantibodies or immune cells that target against our own particular cells or tissues. So this is a very dangerous condition where our own protector tries to kill us. So this is again very dangerous condition. So if you see the immune system disorders, the autoimmune diseases, there are multitude of autoimmune diseases. There are about 100 different types of autoimmune disorders, which are either, the organ specific, which are more systemic. Organ specific in the sense that they are specific towards a particular organ. If we say systemic, means they are generalized, they are everywhere. You think of it like, for example, if you talk of rheumatoid arthritis, it is there in the joints. So it is going to affect all the joints which are there in our body, whether it is relating to the smaller joints in our fingers, the knees, the elbows, everywhere. So in that case, autoimmune diseases, they become very alarming and very dangerous to us. If we talk of the brain, diseases like multiple sclerosis, autism, they are again the autoimmune diseases. The blood, like leukemia, lupus erythematosus, hemolytic dysglycemia. GI tract, celiac disease, Crohn's disease, ulcerative colitis, diabetes type 1. These are all the autoimmune diseases of GI tract. The nerves, that is the peripheral neuropathy, diabetic neuropathy. If lungs, then the fibromyalgia. Then skin, psoriasis, vitiligo, eczema. Muscles, the rheumatoid arthritis. Again, it is there in the bones also, rheumatoid arthritis. Then ankylosing spondylitis, common in both the bones and the muscles, and the other forms of rheumatica. So in a way, we can say that the immune system can act in different ways. If there is too little of an immune response, it is known as immunodeficiency. If it is more of it, okay, then it is known as hypersensitivity. And if it is misdirected, means there is no fine line between the self and the non-self. An immune system starts to work against the self also. Then it becomes misdirected and leads to a condition called autoimmunity. So another way of representing the autoimmune diseases, that is where any virus or any intruder, when gains an entry, immune system comes into play and tries to fight against it and extrude it out of our body. But when we talk about the autoimmune disease, it creates the antibodies that attack your own cells. So foreign system, this might be because the foreign substance resembles our bodily substances, which is under the molecular mimicry that I've told you. Then another way, that is the normal body cells, they become altered because of these kind of changes. Might be because of the genetic predisposition, the environmental factors. There could be many. And lymphocytes sometimes malfunction and make abnormal antibodies. So all these conditions, they lead to the autoimmune diseases. Now, coming to the prevalence, if you'll see, most of these autoimmune diseases, they are actually the chronic inflammatory conditions, where the inflammation becomes so prolonged that it goes out of control. Second in this is that it is about, if you'll see here, about 5% and the 5 to 7% of adult population is affected by these autoimmune diseases. And women are more affected with this case because 2/3 of the cases are more there, like female are more affected with the autoimmune diseases as compared to the male. And this might be due to the genetic predisposition, because it is there in the literature that the X chromosome is a major factor that carries the genes for causing these autoimmune diseases. So might be this is the region, might be some hormonal factors are involved. Second in this, and the last one is that it actually involves there are more than 40 distinct human diseases that originate from the autoimmune dysfunction. Now coming to the, why is there the need to go for the conventional and the targeted system? See, conventional, we know that we give it once and it gets distributed. Like if you can see this figure, you can see that there is a tumor is there, and this is located at this region. So if we give something which is non-directional, it is going to be distributed to our entire body. Okay, so it won't be the targeted. It is a conventional kind of system. But if we give the targeted system, it's going to be targeted at this particular area, and which is actually the thing of interest, which is actually needed. So the peaks and troughs which are there in the case of conventional delivery system, they are being eliminated, and there occurs a controlled delivery in the case of targeted systems. So which is actually the need of the hour. So second most important thing in designing any kind of system is overcoming the biological barriers. See, the moment, these nanoparticles or any such formulation, it enters into our bloodstream, it has to surpass number of barriers which are there. See, I've told you that these macrophages, we can say that a group of serum proteins, they remain in circulation in the entire blood, and they are in actually lookout for these kind of intruders. So if you give the drug also, or the formulation of your drug also, they are easily recognized by these these phagocytes or the serum proteins, and they form a crown onto its surface, forming a biocrown, or we in terms of biotechnology we say it as the this process is known as opsonization, where these opsonins, the serum proteins, they adsorb to the surface of nanoparticles or any particle thereof, and they are then now easily recognizable by the immune system. So they are then extruded out. So first, if I say that if anything has to be a pathogen, it is not easy to be a pathogen, because that pathogen also has to establish itself within our body and has to overcome the immune system. In the similar way, if anything is to be a cure or a therapy, again, it has to overcome our immune system in some way or the other, so that it could establish itself and could lead to the cure of the disease. So once this, within 30 seconds, these serum proteins, they absorb onto the surface of these nanoparticles, and they are then extruded out of the body. They are endocytosed or exocytosed and then cleaved off. But in case of immune disorders, where we actually want our drug to be delivered to the immune site, then in that case it becomes helpful to us. The third thing which is there is actually the interaction with the biological systems. So there are a lot of, the number of the interactions, the protein interactions with the cellular components of the membrane, inside the membrane. Then there is a cellular adsorption at the next level. Finally, the uptake and the recycling of these components, the safety evaluation, and then their further biodistribution and the degradation in the body. So when these conjugated, the systems, the formulation systems, when they absorb onto the cell surface, they are being taken up in a different ways. There are, at the cellular level, there are the clethrine and the cavular-mediated pathways, whereby these are specifically taken up by the process of phagocytosis or pinocytosis, depending on its configuration, and they are then internalized inside the body, digested or acted upon by the body component. So if we say it, if we talk about the delivery strategies, the concept of the clever drug delivery system comes into play, where it consists of the three major moieties. The one is the targeting moiety, right, which is at the outer end, the carrier, which carries the drug, and the major drug, that is a therapeutic drug, which produces the action. So what could be the different components which we could use for this purpose? It could be antibodies, that is the molecules that bind to antigens. It could be proteins. These are the complex molecules with the diverse function. It could be lipoproteins that transport the fats through the bloodstream. It could be hormones, which are actually the chemical messengers regulating the bodily functions. It could be the charged molecules. That is, the molecules with certain charge, whether it is a positive or negative, and depending on the drug molecule, they could bind with it. Then actually, and there also it happens that there are certain tumor sites which particularly are positive for the charged molecules. So we can use these kind of charged component in that case. Then the polysaccharides, these are the complex carbohydrates which are made of sugar molecules. Again, these could be used for targeting purpose. The low weight ligands, which could help to bind these the therapeutic drug carrier to the targeting these small ligands to the target sites. So I'm going to specifically emphasize on the targeting of macrophages. As you can see, this is the structure of macrophage, structure with the extended, the hands to engulf the foreign particles. So in response to the presence of microbial products, the stimulated lymphocytes or damaged cells, these macrophages, they differentiate into the distinct functional populations. So there are certain subtypes of these macrophages. So these are the different locations where these macrophages are there, and they are known by these different names. So if you're aware of it, this could help you in targeting these particular macrophages. Like if we talk about adipose tissue macrophages, they are the cell name is again the adipose tissue macrophage. In the bone, you can target the osteoclast, bone marrow and blood, the monocytes, central nervous systems, the microglia. connective tissue, the tissue macrophages leading to giant cells, the granulomas, the cell name epithelioid cells, and so on like that. Then coming to the macrophages, see, all the cells, they originate from a single, the hematopoietic stem cells. So they then further on differentiation forms the different kind of cells. The one of such is actually the monocyte, which on further differentiation forms the macrophages. Now there are two major subtypes of these macrophages. The one is a M1 subtype, and the one is a M2 subtype. So, the action of these M2 and M1, they're completely contradictory to each other. If I say that I'm going to target this particular drug to a macrophage, that won't serve the purpose unless we are sure that we are going to target the M1 or the M2 macrophage. Because if we talk about the M1 macrophage, it is pro-inflammatory in nature, means it supports the inflammation. It leads to, the increased production of the reactive nitrogen and oxygen intermediates and induction of they're responsible for induction of Th1 response. So in a way, they are, they support the anti-cancer, anti-tumorous activities. Second, the M2 macrophages, they are actually anti-inflammatory in nature, and that is why they are involved in the tissue remodeling, immune regulation, tumor promotion. And so again, this is promoting the tumor. So this is not supportive in case of the tumorous sites, and efficient phagocytosis. So this is again the balancing the macrophage roles in the tumor immunity. So you can see M1, which are involved in the anti-neoplastic immunity. These are pro-inflammatory in nature, whereas when we talk about the M2 macrophages, they are pro-tumor, they exhibit the pro-tumor properties, and they are anti-inflammatory in nature. So again, see a macrophage, but the next question arises that how these macrophages, they succumb to two different pathways. How do they form the M1 and M2? So the triggers are different. If the macrophages, they get triggered with the interferon gamma or lipopolysaccharides in the presence of Th1, they form the M1 macrophages, and then they serve the purpose of pro-inflammatory, releasing certain cytokines like INOS, TNF alpha, IL-1, 6. These all are actually inflammatory in nature, and they are then associated with inflammation, tumor resistance, and graft rejection. Then coming to the macrophage, the M2 macrophages, they are, formed on triggering by the IL-4 and IL-13 with the Th2 pathway, and they then actually form the anti-inflammatory support system because of the release of IL-10, agenase 1, CD206, TGF-beta, VEGF, and so on. So in this, M2 activation is associated with immunoregulation, matrix deposition, remodeling, and graft acceptance. So if we want to go for the if you want to go for a transplantation, now you are aware that which macrophage needs to be activated and which macrophage needs to be suppressed. Then there are certain strategies by which we modulate the macrophage. So one of that strategy is macrophage re-education. Macrophage re-education means we must be aware that which macrophage we need to target, whether it is M1 or the M2. And there's sometimes it also happens that M1 gets converted to M2. It's a constant polarization and repolarization process that takes place in our body, whereby M1 sometimes gets converted to M2 and M2 gets converted to M1. So again, this is an important part of the therapy, whereby the M1, in case of the wounds where the tissue injury and the repair of tissue injury needs to be done. So in that case, M1 needs to be converted to M2. And if the tumorous site is there, the M2 needs to be converted to M2. So again, this is one completely different approach is there. Next is the macrophage depletion. So there are certain, actually strategies which are used, which is done to deplete the macrophages. Third, which is most commonly used, is a nanoparticle-based therapy, where we target these macrophages, activate them, deliver the drug of the choice to the target site. And the last one is the macrophage modulation. That is depending on the kind of, the macrophage action that you want out of it. So targeted delivery operates on two main mechanisms. First, the affinity between the target receptors and the drug carriers for precise drug localization. This is very important. Second, which is very important step, is the strategies like surface modification to enhance the targeting delivery. So these two aspects must be known to you before finalizing any kind of targeted delivery system, specifically dealing with the macrophages. So when we talk about the nanoparticles and nanoconstructs, there are certain parameters which must be taken into consideration. First is its size, second is shape, third is its charge. So if we talk about the size, in general, if the size is too less, it is actually filtered out by the kidney and by other mechanisms. Then if the size is too big, means like here you can see if it is more than, the 2 microns or like that. So it leads to, again, the easy uptake by our RES system, and then it is thrown out of the body. So nanoparticles should be designed in such a way that they must fall within this window of 200 nanometers, like that. Similarly, if we talk about the shape, as I've told you that all the components in our body, they have different shapes, and the shapes actually justify that which kind of work they have to do. Because if we'll see the spherical nanoparticles, they'll be easy to transport from one region to another. They will be easy to move. So their uptake is much easier when we compare it with the rod-shaped structures and further with the disc-shaped structures. So again, the shape is an important parameter while making any kind of nanoconstruct. Then final, which is most important, is the charge on that. Charge on the surface of that nanoconstruct, whether it is negative, neutral, or positive. So the positive ones are taken up, their cellular uptake is even more higher when compared with the other charged particles. This might be because the lipopolysaccharide present on the membrane, they are negatively charged, and they just take up these positively charged components in a better way. So there are different kinds of, the macrophage targeted systems. Either we go for the macrophage targeted delivery system, where we simply do the modification on any carrier system. We attach the ligands which are specifically active against the any kind of macrophage that we want to target. They absorb onto its antibody molecules or the receptors present on its surface and produce its action. Another is the macrophage based delivery system, because anything which will be considered as self by our body, it will be more acceptable to our body. So in this, the first category is a living macrophage delivery system, whereby macrophages are actually used as a carrier molecule to deliver the drug. In this second one is actually the macrophage membrane-coated delivery system, whereby the membranous components of the macrophages are used to deliver a particular drug. Third category under this is the macrophage secreted drug delivery systems, whereby the secretions of macrophages are actually used for carrying the drug of choice. Then the macrophage extracellular vesicle membrane coated delivery system, where the outer vesicle membrane is taken up, extracted, and then it is used as actually the delivery carrier. So these are all the different kinds of delivery systems that have been made using the macrophages. We are more concerned with the macrophage targeting, where the major concept, two major things are taken into consideration. The first is the targeting the macrophage receptors. There are multitude of receptors which are present on the surface of these macrophages, which we can use to target the specific kind of receptors present on the macrophages. Or we can go for targeting the specific macrophage enzymes. We could do the modification on these enzyme fragments so that they're easily taken up. There would be a selective hydrolysis, and then the further targeted action would be there. So talking about the macrophage receptors, the common ones are actually the sialoadhesion receptors, which are there on macrophages, which these receptors, they bind to the sialic acids, which mediate the cell-cell interactions. Second are the folate receptors, and these are the most commonly worked on receptors. They bind the folic acid and mediate its uptake into the cells. Third are the galactose receptors. They bind the galactose sugars, facilitating the cell adhesion. So it could remain adhered to the macrophages for a longer period of time. Then comes the mannose receptors. These receptors recognize the mannose residues on glycoproteins and glycolipids. Then comes the beta-glucan receptors. These receptors, they bind to beta-glucans, triggering the immune response. Then comes the scavenger receptors. Again, these are the most studied receptors on the macrophages. These receptors bind and internalize the modified lipoproteins and other molecules. And then comes the tufstin receptors. Again, these receptors, they bind to the tufstins, which are actually a tetrapeptide kind of a structure, and do the internalization of the drug components. So these are just a symbolic way to represent that how many multitude of receptors are there on the surface of macrophages, which can be used on for targeting the drug of choice to these receptors. And it also happens that in a particular kind of a disease, some of the receptors are upregulated as compared to the others. So if we are aware of it, that which all receptors are upregulated in a particular disease, we could easily mark them and target our drug of choice with onto those receptors. So this is a general overview that how it actually happens, of what kind of work has already been done, the various strategies whereby these nanoparticles have been packaged with the therapeutic agents to target the M1 or the M2 macrophages. So you can see over here the M1 macrophage, it is depleted if we don't want the pro-inflammatory kind of response. So based on the kind of drug that is entrapped, they are depleted, subjected to the apoptotic pathway where there occurs the programmed cell death. This is mediated by the T regulator cells, or it could lead to it is re-educated to form or it is polarized to form the M2 macrophages, so that it would produce the anti-inflammatory response. Then another work is actually on the targeting of specific macrophage enzymes, where a common work by Needham et al., they have worked on it, and they identified a particular amino acid motif which was selectively hydrolyzed by these enzymes, and then it actually helps to target a particular kind of a macrophage. This is another approach which is more chemistry oriented. Now, this is the some of the work that we have been carrying out, whereby we have, we are principally working on targeting the folate receptors, where we have used the superoxide dismutase, which is an antioxidant enzyme, which was absorbed onto these antioxidant mimicking nanometrics frameworks for the treatment of rheumatoid arthritis, where the core had the antioxidant ceramic core was there, onto which surface the folate conjugated SOD were absorbed, and we really found a good results with the in vivo studies. And another work, the hypothesis that we have worked on again targeting these RA the folate receptors, where we have used, instead of these enzymes, we have used the glucocorticoids and targeting them onto by means of the folate receptors. And this further activated the IROM2 and TNF alpha pathway, and release of the further cytokines, which are further anti-inflammatory in action and further supporting the anti-rheumatic effects. So if we talk about these systems, these success stories are really very meager. And these targeted nanoconstructs, only few have been available in the market, but not for autoimmune disorders still date. So but the another ones like pegylation has been really vastly studied, where the PEG molecule is adsorbed onto the surface of these nanoconstructs, and they have been successfully launched in the market in the name of various brand names, and they are actually serving as a major player in various diseases like Crohn's disease, the multiple sclerosis. But there are certain drugs which are there for the autoimmune disorders, which are in the clinical trial stage, like type 1 diabetes. It is in the phase 1 trial using the gold nanoparticles. For plaque psoriasis, again, a phase 1 trial is going on with the uncoated nanoparticle ointment containing paclitaxel. Then in the rheumatoid arthritis, a phase 2 trial is going on with the liposomal prednisolone, and phase 3 is investigating pegylated nanomolecules as the TNF alpha inhibitors. So, the work is going on. And but again, there are a lot of challenges which are to be addressed. So first in this is that research must address the challenges like nuclear localization and cytotoxicity when we are working on the nanoparticles, because we talk about the nanoparticles. If its size comes out to be less than 10 nano, then it might happen that they might gain entry into our nucleus and might produce the the genetic inheritable changes. So all these issues need to be addressed while actually designing any kind of nanoconstructs. Then there must be proper optimization of delivery timings, which are based on the macrophage infiltration dynamics in disease. second most important is that there is a heterogeneity of macrophages. It must be known to you that there is which stage is required for your the drug delivery, whether it is M1 or the M2 phenotype. So nanoparticles must be engineered in accordance with that. Then comes the complex interactions between the nanoparticles and cells. One example I would like to quote over here is that the physiological fluids also, affect our interaction of nanoparticles with our biological systems. Because, if we talk about any nanosystem, I've told you that once they gain entry into our bloodstream, immediately the serum proteins, they come into play, they absorb to it, and they are then, they undergo the process of opsonization. But there are certain biological fluids, like in the case of inside the liver cells. There, the mobility, the turbulence which a nanoparticle faces, as compared to when it is outside that particular organ, it is completely different. And then the kind of the results that we are left out with, they come out to be really different. So these things need to be kept in mind while preparing the nanoconstructs, especially for addressing such physiological systems like macrophages. So I would say the research search is always on, and a lot of work needs to be done. A lot of questions needs to be answered, and this was just an overview that I've given, that in various ways we could target our immune system and could address the various issues of immunity. And this is my research team, and whatever little we are working on is because of the combined effort of my team, who are working diligently on various topics, various research areas. So it's basically my team. I'm thankful to. My kind acknowledgement to my mentor institute, my University Institute of Pharmacy at Pandit Ravishankar Shukla University, the School of Pharmacy, where I really learned so many things while working there on proteomics and various other methodologies at University of Mississippi, Oxford. My granting agencies for providing me with the funding like UGC, ICMR, DST, DHR, and so on. So with this, I come to the end of my presentation. Thank you so much.
Thank you so much, ma'am, for delivering such a comprehensive and insightful session on targeted nanoconstructs for the treatment of autoimmune diseases. Your expert discussion on advanced nano-carrier system, their therapeutic relevance, and potential in managing complex autoimmune conditions was truly enlightening. The clarity, depth, and real-world applicability of your presentation have greatly enriched today's session and inspired deeper interest in this critical area of pharmaceutical research. We sincerely appreciate your time, expertise, and valuable contribution to the 95th SPSR webinar. Thank you so much, ma'am.
Thank you. Thank you so much.
With your permission, ma'am, we will now take some questions from our participants.
Sure.
So the first question would be, what are the key immune system triggers that initiate autoimmune responses, and how can they be therapeutically targeted?
See, if we talk about our immune system, there is no single thing which is responsible for it. There are multitude of triggers, be it, the genetic predisposition, our genes. You must have seen that some people are highly sensitive to or susceptible to a particular kind of immune disorder as compared to others. So genetic predisposition is there. There are environmental factors which are, again, responsible for some kind of, these are also the kind of triggers. If we talk about the therapy, so there are a number of multitude of therapies are there, be it the immunomodulators, the working in this area. We could really target the various, if the cause is known to us, we could really, if triggers are known to us, we could really address the issues relating to these autoimmune disorders.
Thank you, ma'am. Let's take one another question. How do M1 and M2 macrophages subtypes influence the progression or suppression of autoimmune diseases?
See, I've told you in my slides also that M1 and M2, these two subtypes of macrophages, they work in the opposite way. Like if the M1 progression takes place, if the M1 macrophages are formed, they are more pro-inflammatory in nature as compared to the M2, which are, anti-inflammatory in nature. So if the M2 macrophages gets triggered, they lead to the condition whereby, the the more of the inflammation condition comes into play. These are more of like tumor controlling things. But when we talk about the M2 macrophages, they are anti-inflammatory in nature. They are more so involved in case of your tissue repair, the injury, and so on. Especially in case of wound healing, they're very important.
Thank you so much, ma'am, for this answer. So another question is there from the participants. What are the major advantages of using targeted nanoconstructs over conventional drug delivery systems in autoimmune therapy?
There are a lot of, the benefits of targeted delivery systems over the conventional systems. Be it if we talk about the efficacy, obviously, there are two things which are very important when we decide any system. The first is the therapeutic efficacy, and the second thing is the toxicity. So any delivery system that we are working on, it must improve the efficacy of the drug. It must improve the bioavailability. It must improve its effectiveness in a way so that the dose can be reduced. So this is the first thing which is there in the case of targeted delivery system. The second thing, that is the toxicity, obviously, because we are targeting our drug moiety towards the target site. So obviously the toxicity gets reduced. So this is another thing. Third thing, the barrier, the biological barrier, which is again an important issue with the case of the delivery system. These all three things are actually addressed upon in the case of the targeted delivery systems.
Thank you, ma'am. So one last question, and it's really interesting. So what are the biggest challenges in translating macrophage targeted nanocarriers from lab to clinical settings?
See, challenges are too many, because if we talk about, our nanoconstructs, it's a complex system. If we talk about the immune system, it's a complex system. So altogether, these two things are so complex. And when we talk about the scale up, the industrial, the further scale ups of the things, it needs to reproduce the things in such a way that the similar results are obtained at every time. So I think if you ask about the scalability approach, this is again very challenging based on the multitude of the diversity, whether we talk of the nanosystems or we talk of the immune systems. All the work is going on, and we are hopeful that we are going to get something positive out of it.
Thank you so much, ma'am, for addressing all the questions with such clarity, depth, and scientific insights. Your thoughtful responses have added great value to today's discussion and helped us better understand the intricacies of targeted nanoconstructs in autoimmune disease management. We sincerely appreciate your time, expertise, and the engaging way in which you shared your research. Thank you once again for your outstanding contribution. So before we conclude, I really thank you for contributing to the 95th SPSR International Webinar, ma'am.
Thank you so much. Thank you, Ms. Rupam, and the entire team of SPSR for giving me the opportunity to interact with all of you. Thank you so much.
As we conclude the 95th SPSR webinar, we extend our sincere gratitude to Dr. Manju Rawat Singh for her enlightening and impactful session on targeted nanoconstructs for the treatment of autoimmune diseases. Her presentation provided deep insights into the design, development, and therapeutic applications of advanced nanocarrier systems for targeted immunomodulation. The session emphasized the growing importance of site-specific drug delivery, the role of macrophage target therapies, and the translation of innovative nanoformulations into meaningful interventions for autoimmune disorder. We are confident that today's discussion has sparked new ideas and encouraged further research collaborations in the field of pharmaceutical nanotechnology and immunotherapy. On behalf of the Society of Pharmaceutical Science and Research, SPSR, we sincerely thank Dr. Manju Rawat Singh for her valuable contribution and all participants for their active engagement. We are deeply thankful to Dr. Shashi Alok, the National President of SPSR, for honoring us with his presence. We recognize and appreciate your commitment and guidance, and we offer our profound thanks for your significant input. We would also like to express our appreciation to our convener, Mrs. Monika Sabharwal, for her pivotal role in organizing this event. We extend our gratitude to all SPSR team members for their collaborative efforts, which has broadened our understanding and fostered comparative learning. Your collective contributions have made this webinar a success, and we look forward to continued collaboration in advancing pharmaceutical sciences and research. We look forward to your participation in future SPSR webinars as we continue to explore groundbreaking topics and foster knowledge sharing in pharmaceutical sciences. Thank you once again, and we look forward to welcoming you to our upcoming webinars. Stay inspired, stay innovative. Have a wonderful day. Thank you.